Gosh, I don't normally speak to pharmacologists. I'm a gene therapist. This is a little bit like walking into the lion's den. But I'm interested to get your feedback and opinions, because as gene therapy and gene editing are becoming clinical realities, we actually do need to sort of engage your community in helping us to think about this. These are my disclosures. And this is just an outline of what I want to do in the next half an hour or so. Quickly just pitch to you why I think HIV is a candidate for gene editing. Give you some sort of gene editing basics. I'm assuming some of you might not even know what CRISPR is. Talk about, at a high level, some of the opportunities that might be available for using gene editing against HIV, and then finally wrap up with some speculation about what might be happening in the next 20 years, or given the pace of gene editing, quite possibly the next 20 months. Okay, so the sort of fundamental thing that occurs to me when I think about HIV and the challenge of why antiretroviral drugs can't cure the virus is, of course, because of this tendency the virus has to integrate into the genome and in some cases become transcriptionally silent or latent. And that would be fine apart from the fact that this latent HIV can subsequently be reactivated. And so, of course, this means that people need to take lifelong antiretroviral drugs. But the fact that the virus integrates into the chromosome also gives the disease some of the features that we also see in genetic diseases. And this is one of the reasons that HIV infection has attracted the attention of gene therapists. So to give you some basic background about gene editing, the most important thing to understand is that gene editing uses tools that recognize specific DNA sequences. And they can do this in a number of different ways. So what I'm showing you here in this cartoon, on the left-hand side is a schematic of CRISPR-Cas9. So this is a bacterial system that we have hijacked to allow it to be used as a gene editing tool. And the way that it works is there are two components. First of all, the CRISPR, or guide RNA component, is basically a stretch of about 20 or so nucleotides of RNA that you design that are homologous to your DNA sequence of interest. And this guide RNA then brings in the Cas9 protein, which has endonuclease activity. So a CRISPR-Cas9 complex will introduce a site-specific DNA break. On the right-hand side, I'm showing an alternative gene editing tool called zinc finger nucleases. These were a precursor to CRISPR-Cas9, and they do exactly the same thing. They introduce a sequence-specific DNA break. But in this case, the specificity for your DNA sequence comes from these sort of peptides that each bind to 3 bases of DNA. And so linking these into an array gives you a DNA binding platform that can confer specificity. So both of these tools are sometimes called targeted nucleases, and they introduce a DNA break at your DNA sequence of interest. And then what happens afterwards depends on which DNA repair pathway the cell uses to repair that DNA break. So in human cells and mammalian cells, the simplest DNA repair pathway is called non-homologous end joining. And it's kind of like an emergency DNA repair system. The ends of the DNA break are kind of stuffed back together, and this is done in a way that often leads to small insertions or deletions of the sequence at the site of the break, and so it leaves behind a scar or an indel. And of course, the consequences of this are that if you have your DNA break occurring in the middle of a gene, this indel scar can actually knock out the sense of the open reading frame. And in addition, if you had targeted nucleases that recognized an HIV sequence, you can imagine that this could also lead to disruption of the HIV virus. So that's non-homologous end joining, but a more useful pathway and a more complicated pathway are DNA repair pathways based on homology-directed repair. And these pathways, which are much more precise, instead copy DNA information from a template. Naturally, this template would be the sister chromatid. But we can sort of hijack that process by introducing into the cell a DNA template. It can be a plasmid DNA, or it can be a DNA virus or an oligonucleotide. And the information in this DNA template then gets copied. And you'll see that this gives us some possibilities because, for example, your DNA template can actually contain a mutation, a change, or an edit that you want to introduce. And in that way, you can, for example, correct a mutation in a gene that causes sickle cell disease. In addition, you can also use this mechanism to insert larger DNA cassettes, including whole genes and promoters, again, in a site-specific manner. So it's a way of doing gene therapy, but with the added advantage that you can control where your gene is going to go and therefore improve the precision and the safety of the process. And then the final gene editing tool I want to tell you about are a range of different strategies that are based on what's called dead Cas9. So if you remember, I told you that Cas9 is normally an endonuclease. Dead Cas9 has mutations in the enzyme that prevent it cutting the DNA, but still, when combined with a CRISPR guide RNA, retain the capability to be targeted to a specific DNA sequence. And so you can fuse to the dead Cas9 different effectors, proteins that can switch on or switch off a gene or introduce epigenetic modifications. So in this way, dead Cas9 fusion proteins can be used to modulate gene expression without having to introduce DNA breaks. So this table, and I'll use this throughout my talk, sort of summarizes the different mechanisms that gene editing has available, and then some of the potential applications that are being worked on or speculated for HIV. And I'm going to, again, at a fairly high level, give you some examples of how that might work. So I'm going to first of all start off with, if you like, the application that has been the poster child for gene editing, which is using NHEJ repair to disrupt the CCR5 gene and thereby confer resistance to HIV. And the reason that this was so attractive to gene editors is because of the reports, and there are now potentially 3 reports of individuals who've been cured of HIV by receiving a transplant of hematopoietic stem cells, a bone marrow transplant, if you like, from donors who carried 2 copies of the mutated gene CCR5 delta 32. This mutation basically disrupts the CCR5 gene, and these donors, therefore, don't have a functional CCR5 gene and are effectively resistant to HIV, at least the R5 tropic strains. And so these 3 individuals here, who are kind of known colloquially as the Berlin, London, and Dusseldorf patients, each had an underlying malignancy, leukemia, or a lymphoma that necessitated them undergoing bone marrow transplantation. And their doctors were able to identify bone marrow donors who weren't just tissue matches, but also were carrying this CCR5 delta 32 mutation. About 1% of the population is homozygous for this mutation. And in all 3 cases to date, they appear to be in remission or cured of HIV. The Berlin patient has been this way for now more than 10 years. So these sort of, if you like, powerful N of 3 now human demonstrations that loss of CCR5 can lead to HIV resistant was really what motivated gene therapists and gene editors to think, can we recapitulate that using gene editing tools? So some of the challenges we face in doing that are that our technology cannot exactly mimic what happens when somebody undergoes a bone marrow transplant. So for example, when you're receiving an allogeneic, which means from a donor, hematopoietic stem cell transplant, and especially if you are receiving this because you also have cancer, then the cancer treatment itself, the chemotherapy, radiotherapy or immune depletions that ablate your cancer will also deplete other hematopoietic cells in your body and effectively deplete this reservoir of latent HIV that you are carrying. At the same time, because donors are never perfect matches to the recipients unless you're an identical twin, there is always some level of what we call graft versus host disease, whereby the donor cells and the donor T cells can attack the recipient, and that can further deplete the latent reservoir. So these factors are not going to be features of, instead, what we call an autologous hematopoietic stem cell engineering therapy. So these are some of the challenges we face. So my lab's been working on this problem for a decade now. We started off with a simple question: could we take gene editing tools and achieve sufficient levels of knockout or disruption of the CCR5 gene in order to have an effect against HIV? And because we were doing this 10 years ago, this was pre-CRISPR-Cas9, and we were working instead with zinc finger nucleases. And we basically developed a system whereby we could introduce the zinc finger nucleases into human hematopoietic stem cells by electroporation of messenger RNA that expressed the ZFNs. And then we took those edited hematopoietic stem cells and transplanted them into immune-deficient mice. Immune-deficient mice transplanted with human hematopoietic stem cells will actually grow you a human immune system after about 3 months. And so that allows you to challenge the humanized mice with HIV and see whether or not your therapy has had any effect. So this graph shows the results of those sorts of experiments where the humanized mice were challenged with R5 tropic HIV. And you'll see that all of the mice originally became infected, as shown by levels of HIV in their serum. But whereas the mice receiving control untreated human hematopoietic stem cells, the levels of HIV stayed high. For the ZFN-treated mice, they were eventually able to overcome and suppress virus replication down to undetectable levels. And the reason that this happened, or at least the reason that the mice were originally infected, is entirely because the levels of gene editing we were able to achieve 10 years ago, we got excited if we could modify 5% of the cells. So I think what's happening here is, 95% of the cells are wild type and able to support HIV infection. We got a robust initial HIV infection, but then the CCR5 positive cells that could be infected were killed, and our engineered CCR5 negative cells, although initially in a minority, were sort of able to be selected to the point where they were able to suppress the infection. So this has now progressed into clinical trials. And on this next slide, I'm sort of summarizing the current status of clinical trials targeting CCR5. So at the top, this is just a reminder that the strategy of taking people who are candidates for a bone marrow transplant because of cancer and trying to find them a matched CCR5 delta 32 donor, that is still being evaluated, although obviously it's only ever going to be applicable for a very small number of people. And there's a consortium in Europe called ICSTEM that is doing that. And then in the yellow boxes, these are the two clinical trials that are, or series of clinical trials that are using the zinc finger nucleases against CCR5 I just told you about. The very first trials actually edited the CD4 T cells, and then the trial that I've starred is the one that my group was involved in the preclinical studies for, which is to edit the hematopoietic stem cells. And then finally, at the bottom, in a sort of a best of all worlds kind of approach, there's a Chinese group who are taking patients who need to receive a bone marrow transplant, but rather than trying to find them a matched CCR5 negative donor, they are taking the donor cells and mutating those so that they are CCR5 negative ahead of doing the transplant. And that's an interesting approach because it sort of still combines the gene editing, but does it in a scenario where the patients are going to be undergoing the conditioning for their malignancy that might give better outcomes. Okay, so that's the sort of the most famous, if you like, application of gene editing. I should say that these trials were the first times that gene editing had been done in humans. And CCR5 is a rather unique application because there are not many genes in the body where actually destroying the gene is expected to give a beneficial effect. So my group are much more interested in, if you like, the second group of manipulations where we can use the homology-directed repair pathway to introduce more precise mutations or site-specific insertions to have an anti-HIV effect. So first of all, giving you an example of what I mean by site-specific insertions of anti-HIV genes. So here's a molecule we work with. It's called ECD4IG. It was developed by Michael Farzen's group at Scripps Jupiter. And ECD4IG basically combines CD4 with a peptide mimetic of CCR5. And so this sort of combination biologic can bind to viruses, bind to the envelope protein and stop them infecting cells. And so the idea behind this gene therapy strategy is that we actually go into the CCR5 locus, disrupt it with our zinc finger nuclease or CRISPR-Cas, and then use homology-directed repair and a homologous template to site-specifically insert an expression cassette for ECD4IG at that locus. So we're kind of combining two different anti-HIV gene approaches. When we do that and, for example, put edited cells into humanized mice and monitor them over several weeks, you'll see that the levels of ECD4IG we see in the serum of the mice, they can rise to be a mg per mil. And then when we challenge the mice with, in this case, an SCXCR4 tropic virus, because we know that CCR5 works to protect the mice, here we want to ask, does ECD4IG being secreted systemically also give us any effect? And when we challenge the mice with an X4 tropic virus, the mice are all resistant. So what this shows us is that gene editing can be used to create if you like, a factory in the body that can be pumping out this biological molecule that can then protect the mice from HIV infection. And so this sort of application could equally apply to, for example, putting broadly neutralizing antibodies into this locus. Now, the next thing I want to give you an example about is how we could actually use gene editing to engineer protective anti-HIV alleles. So what do I mean by that? Well, the idea behind this is that as people are doing sort of GWAS studies and analyzing people who are resistant to HIV or various types of controllers, the hope was that we would identify some genes, some alleles, some SNPs that could protect people. And obviously, the main one that came out of these analyses was the CCR5 delta 32 allele, but there have also been other alleles, for example, HLA haplotypes. Unfortunately, those are too challenging to edit because I think of all sorts of sort of unintended consequences we might expect. And so I've been a little disappointed by this. And so instead, my group is looking at what we call clues from primate orthologs of restriction factors. So in case you don't know what restriction factors are, I have a simple summary here. It's not very simple, is it? So restriction factors, this is a catch-all phrase for a group of very diverse intracellular proteins that have antiviral or anti-HIV effects. They're shown in yellow on this slide. They are things like tetherin or ApoBec or TRIM5-alpha. And the reason that HIV-1 can still infect us despite all these different defenses is because HIV has developed antagonists of these various factors. So, for example, the VPU protein degrades the tetherin restriction factor. And in contrast, when oftentimes if you take the same gene from a primate, rhesus macaques, it now has activity against HIV-1 because HIV hasn't evolved to be able to infect rhesus macaques. So we can compare the sequences of the human and the primate forms, and we can identify, for example, what amino acids are different between the two, and which might therefore be candidates for us to insert into our human genes. So just giving you an example with TRIM5-alpha of how this works. So TRIM5-alpha is an intracellular protein that recognizes incoming HIV cores. It does this through the terminal exon of the human protein called the spry domain. And when it recognizes the cause, it both prevents HIV uncoating properly and also triggers NF-kappa B in an inflammatory response. And HIV has learned to protect itself from human TRIM5-alpha by coating itself with cyclophilin A. So the strategies we are taking to try and get around this defense by HIV is, first of all, by introducing a couple of mutations that exist in the rhesus macaque form, which can convert the human form into a form that can now recognize HIV-1, and also using a trick from sooty mangabeys, which are my favorite primates by far, we can make something called TRIM-CYP, where we replace this terminal spry domain, which is now ineffective at recognizing the HIV core, and we actually replace that with cyclophilin. So it's kind of like a checkmate move, in that in order for HIV to counteract a human TRIM-CYP protein, it would have to evolve away from its ability to bind cyclophilin, and that would then leave it vulnerable to unedited TRIM5-alpha. So we think having the combination of both the normal gene and a mutated gene, an edited gene, might prove something the virus can't mutate around. And so just showing you the types of experiments we do. Again, humanized mice challenged with HIV. We had very low levels, only about 5 or 10% of the cells edited, but I think you can see over time that the levels of HIV in the control or a GFP control mice are higher than the mice that received either of our two editing strategies. Okay. So next, we can also use gene editing to think about engineering and improving immune cells in the body to recognize either HIV or HIV-infected cells. And in many ways, we're sort of following what's happening in the cancer field with, for example, CAR T cells. So CARs, chimeric antigen receptors, are basically artificial T cell receptors that allow a CD8 cell to recognize cells beyond the way it normally recognizes by recognizing class 1 peptides. And so CARs are often, they comprise the sort of intracellular signaling domain of the T cell receptor, but the binding part on the outside is replaced, for example, with a single chain antibody. And in the case of HIV-1, it is instead, it can be replaced with the ectodomain of CD4, because obviously GP120 binds to CD4. And gene editing is also being used to further improve these sort of first-generation CAR T cells. So, for example, we can use the homology-directed repair pathway to site-specifically insert the CAR cassette at a defined locus. This improves safety rather than using, for example, a lentiviral vector. And it also gives us the potential to insert the cassette at the endogenous T cell receptor locus, so that we both knock out the endogenous receptor and we also have a cassette that is now regulated in the same way that the T cell receptor is naturally. In addition, we can also think about knocking out other genes whose function interferes with the function of a CAR T cell. So, for example, molecules like PD-1, which lead to T cell exhaustion, if we knock those out, we can extend the functionality of the CAR T cells. And then I think excitingly, we can also use gene editing to get towards what we call universal allogeneic or off-the-shelf reagents, meaning that you don't have to take a patient's own T cells and engineer them to recognize a cancer or to recognize an HIV-infected cell. Instead, we can take T cells from one donor and make enough reagent for many patients. And to do this, we would knock out both some component of the HLA molecule so that the engineered T cell no longer looks to be foreign to the patient's immune system. And conversely, by knocking out the T cell receptor, we can also prevent those T cells from attacking the host through graft versus host. And the little schematic, I stole it from the website of one of the companies working in this space, Sangamo, just showing that you can knock out both HLA and the T cell receptor, which is CD3, at very high efficiency. So I think this is likely to be a real reality very soon. In addition, my group is working to edit the immunoglobulin locus. We are able to insert broadly neutralizing antibodies into the immunoglobulin locus. And not only does this give us a way to secrete these expensive biologics in a person, but because we're putting this antibody into the immunoglobulin locus, we anticipate that that antibody will be able to continue to evolve. It will respond to the presence of its antigen, and it will continue to evolve through somatic hypermutation. So we're really trying to improve the immune response, at least the humoral immune response against HIV, by starting from a good broadly neutralizing antibody. And then the final example I just want to touch on is that we can also think about disrupting integrated latent HIV. Again, I've already told you about the example where we could have nucleases that disrupt the integrated virus, and then with the dead Cas9 effectors, we can also think about either shocking, blocking, or locking the latent HIV in some way. So mimicking, a lot of things that are being developed also with drugs. And here's just a little cartoon showing what that might look like if we have nucleases that recognize the LTR. Not only can they disrupt the LTR, they can also drop out the whole cassette. And the graph on the right shows how we are able to reduce the amount of reactivatable HIV, latent HIV in splenocytes we take from humanized mice. Okay, so in the last couple of slides, because the talk and this session is about the next 20 years, I thought I'd try and speculate a little bit. So the first question, of course, is this even appropriate for HIV? I'm talking about a complex gene and or cell therapy for a disease you can treat with drugs. What about safety? We really don't yet have the clinical experience to know that we can do gene editing safely. Can gene editing be a one-time treatment? This would be our hope. Then you don't have to worry about adherence. I'm going to put a lot of you out of business, right? But monitoring that presents huge challenges. And then, of course, there's the aspect of public acceptance, and things like the CRISPR babies from China that I'm sure you heard about a few months ago certainly don't help us with that. And so my final slide is just, again, by being at this meeting, I started to think about CRISPR-Cas as a drug. Several gene therapies are now approved and licensed by the FDA. Cancer CAR T cells, because that's proving so effective and so exciting, that's really driving innovation in terms of manufacturing capability. But for us, delivery remains the main challenge. Do we have to edit cells ex vivo, which really limits us to hematopoietic cells? Or can we do in vivo delivery through, for example, nanoparticles or viral vectors, where the specificity of delivery is still the big challenge? And then there's also a lot of conversations now happening around how the heck do we price this? Is this a value equation? Do we have an upfront price versus a sort of a subscription price to defray the high costs involved? And is pricing linked to outcome? It's a bit like having Netflix in a way. You only pay for it if it's working. And for those of you who are interested, I'll draw your attention to there was a position statement came from the American Society of Gene and Stem Cell Therapy in December talking about these things. Okay, and with that, I'll just recognize the members of my lab, present and past, and some of my major collaborators and sources of funding, and thanks for your attention.